My name is Jenny. My name is Jenny Savino. I have the great pleasure of working in the alumni office, and we're thrilled to have our panel tonight, our Blue Runs Deep series. And I love to open it up and just kind of give a couple of comments of framework, if I may. One, we purposely choose this format as opposed to the webinar format of Zoom so that we can be in company, in family company, if you will, all around the world together. Whether you choose to keep your camera on or not is totally up to you, but I just wanted to explain why we do it in this format. And because of that, if you want to go and see, if you're on speaker view, I have colleagues on my team who will make sure that you have the best experience that you can by being able to see the speaker. I'm probably enormously on your screen right now, but that's by design, so you can hear the speakers when they actually come on. Secondly, as you'll see under my name and some of the other names, we encourage you to consider renaming yourself to share what class you are and whether it's your own class or parent years. And you can do that by going over your picture in the top right corner. There are 3 dots, and it gives you the opportunity to rename and add your class years. If that's a tricky thing for you to figure out, just open the chat. You can chat me or Meg McGuinn or Naomi Mashburn, and we can adjust that for you. Finally, we keep folks muted just so you can have the best listening experience that you can. And this is being recorded. And then final is that closed captioning is possible. So you can go along the bottom of your screen and turn that on if that makes for a better experience for you. That's the end of my babble. I am now going to turn it over to wonderful stars that we work with, Annabelle Wu. Thank you.
Good evening. Welcome to our alumni, parents, faculty, staff, and students. We are so excited to have you all. My name is Annabelle, and I'm an upper from Beijing. I'm a member of STARS, and I'm here to introduce today's Blue Runs Deep program. The Blue Runs Deep speaker series was launched in 2021 by the Student Alumni Representatives, also known as STARS. We are a group of students interested in connecting with alumni in various ways through programming with the Alumni Office. Today's focus is alumni in scientific research and pharmaceuticals. STARS will pose questions that we have written, and we have also included some questions from attendees who have submitted them as part of their registration. You will learn from our panelists about their professional paths since Andover through the course of this conversation. Many of our questions will be directed towards a single panelist, but we encourage panelists to elaborate on others' responses. We only have an hour together, so we'll jump right in. To introduce our panelists, I would like to welcome Connor from the class of 2025.
Thank you, Annabelle. Hello, everyone. My name is Connor, and I'm a senior from Southborough, Massachusetts. Our first panelist is Dr. Andrew Ahn from the class of 1982. Dr. Ahn graduated from Columbia College with a Bachelor of Arts in Philosophy and Biochemistry. He then returned to Boston, where he completed an MD PhD fellowship at Harvard Medical School, where he focused on neurobiology, and also worked with the Genetic Screening Study Group to explore the ethical, legal, and social implications of the emerging genetic technology with the media and society, leading to the U.S. Genetic Information Non-Discrimination Act. Dr. Ahn is trained in neurology at the University of California, San Francisco, where he obtained funding from the National Institutes of Health to start a laboratory, and later moved his research program to the University of Florida. Dr. Ahn then went on to lead clinical and scientific drug development teams at Lilly Research Laboratories and Teva Pharmaceuticals for a range of neurology, psychiatry, and pain indications. Currently, Dr. Ahn serves as a Senior Vice President of Clinical Research at Alnylam Pharmaceuticals, which has been a leader in developing a novel class of RNA-based gene-targeted therapies for diseases of the central nervous system. We welcome Dr. Ahn here tonight.
Hello, everybody. My honor to be here. Thanks.
Thank you, Connor. Hello, everyone. My name is Orla Naughton, and I'm a senior from Andover. Our second panelist tonight is Zahra Bhaiwala from the class of 2010. Ms. Bhaiwala received her Bachelor of Science in Biological Sciences and Middle Eastern Studies, also from Columbia University, and then went on to attain her Master's in Health Economics from the Harvard T.H. Chan School of Public Health. Ms. Bhaiwala is the Director of Health Economics and Outcomes Research at Vertex Pharmaceuticals, leading market access and reimbursement support of their cystic fibrosis and type 1 diabetes portfolio in North America. She has a deep interest in creating access to high-cost therapies in low-income markets. Ms. Bhaiwala is also a board member and former chief operating officer of Hikma Health, a nonprofit startup out of the MIT Refugee Accelerator working to bring healthcare data solutions to clinicians treating refugees, and also a member of the World Economic Forum's Global Shapers. We welcome Ms. Bhaiwala here tonight.
Hi, everyone. Looking forward to getting to know you all.
Thank you, Orla. Our third panelist is Dr. Donald Ganem from the class of 1968. He received his bachelor's and medical degree from Harvard University. Dr. Ganem is a virologist and infectious diseases specialist whose research has focused on the hepatitis B virus, Kaposi's sarcoma-associated herpes virus, and the search for new viruses in human and animal diseases. After subspecialty training in infectious diseases at the University of California, San Francisco, he ultimately became a professor of microbiology and immunology and medicine at UCSF, and an investigator at the Howard Hughes Medical Institute, serving there for over 25 years. Dr. Ganem then went on to become the global head of infectious disease research at the Novartis Institutes for Biomedical Research, where he led teams developing novel antivirals for several viral infections and antibiotics for multi-resistant gram-negative bacteria. Dr. Ganem is now the co-founder and CEO of Via Nova Therapeutics, a new biotech firm specializing in therapies for viral infections that have been neglected by Big Pharma. He is a member of the National Academy of Sciences, the National Academy of Medicine, and a past president of the American Society of Virology. We're honored to have Dr. Ganem with us here tonight.
Very glad to be here.
Thank you, Annabelle. Our fourth panelist is Dr. Marianna Kleyman from the class of 2003. Dr. Kleyman received her Bachelor of Science from McGill University in Microbiology and Biotechnology and her PhD from Dartmouth College in Biochemistry. She has held jobs in cancer biology and immunology as a scientist for MIT, Harvard Medical School, and Bicycle Therapeutics. Dr. Kleyman started at AbbVie as a bench scientist, then ran her own lab, and now oversees the work of multiple labs in support of clinical trials. Dr. Kleyman currently serves as the principal scientist in precision medicine at AbbVie and is focused on using her knowledge of human biology and the immune system to guide clinical decisions for new treatments of diseases such as ulcerative colitis, Crohn's disease, rheumatoid arthritis, and hidradenitis suppurativa. We're thrilled to have Dr. Kleyman as a part of our panel.
Hi, everyone. Thank you. I'm really happy to be here.
Thank you, Orla. We'll kick off with our first question to Dr. An. You are clearly a scientist grounded in human nature, having studied the ethical, legal, and social implications of genetic technology, and majoring as an undergrad in both philosophy and biochemistry. This approach must impact the work you do today, leading therapeutic research at a giant pharmaceutical company. How does your moral compass, your VP role at a for-profit global company, and your desire to make innovative treatments for people who need your help drive you to do your work, or possibly even conflict at times?
Yeah, Connor, thanks for the question. I think that there are both moral and practical trade-offs in everything we do. Just, whether you take Route 93 on your commute or not, or whether you stop by to get a cup of coffee on the way. I think there are all those choices that take place. They are only occasionally really ethical ones. But I think that there are some, in the news is really big conflicts posed between serving the needs of your patients and serving the needs of a shareholder or the company's goals. In truth, I see my role as a leader in that process is really not really so fraught by that. I think that I come to work every day really experiencing myself really as a person leading a therapeutic area focused on neurologic disease. I come to work every day principally to bring the voice of the patient to the room. And that's typically involved in when I'm trying to think about how these abstract genes and these abstract targets that we think we can target can lead to some kind of benefit in some kind of neurologic disease. Sometimes that relationship between the gene or the target and that disease is not at all clear. And sometimes that disease has a lot of different dimensions to it. You have changes in cognition, changes in the way you think, changes in the way you get around and move. And the question is, how do you measure clinical benefit in that setting? And so in a lot of ways, the real hard part is really understanding what the patient wants and needs, and then bringing the science to bear to be able to measure those outcomes, and then connecting them with the science of what the target does, what it does in your cells of your body, what it does in the regulation of neurologic function. There are, of course, diseases that have some real challenges in being in first making a medicine for that disease and then having it be profitable. And I think that it's something that maybe Zahra could bring out a little bit more, and being able to really identify how those benefits have to be really quantified in some way. And fortunately, I do have counterparts like Zahra in all the organizations I've worked in who helped me work on those things. And how do you bring that message of real value, economic value, so that payers and other insurance companies, other folks can really understand what the value of that medicine is. I hope that gets as far as we need, Connor. I'm happy to elaborate further.
Thank you, Dr. An. Our next question is for Ms. Bhaiwala. Based on your experience, what are specific ways for healthcare to become more accessible in areas of the world which are considered less developed? What are the biggest challenges in bringing high-cost therapies to low-income markets? And when it comes to promoting access to high-cost therapies, is it more effective to simply decrease the cost of the therapy, or to focus more on securing funding and raising awareness for that therapy?
First of all, I love that you asked this question. I doubt we'll solve it with just me here on the Zoom. I think a lot of smarter people smarter than me are thinking about it. I'm certainly thinking about it. Maybe I'll start with just to kind of humanize the question. I think while I was in high school, while I was in college, before I got much exposure to working in biotech and pharma, I think I would be one of those people out there with a picket sign being like, why doesn't company X, have this medicine in this country in Africa, and just kind of be blaming it on the high prices. I think now that I've spent a lot more time in biopharma, I have a lot more appreciation, not just for the drug development process and how long and expensive that is. I'm sure people are familiar with that sort of language, but I think there's a lot more that goes into, especially thinking about the high cost rare disease therapies. There's a lot of challenges that I didn't realize until after I sort of started working. So I'll just discuss a few of them and maybe just give my thoughts on sort of some of the solutions that we've seen in the industry, and then kind of, I think, where there aren't as many examples to point to, and we're at hopefully a really interesting juncture in the industry, and specifically at Vertex. So maybe I'll try to summarize some of that real quick. So obviously, from an extreme drug cost perspective, some of the gene therapies that Andy might be working on, or others, similarly at Vertex, some of these are anywhere from $100,000 for some of our CF pills, $300,000 a year, every year for the rest of your life, to $2-3 million per treatment for a gene therapy. That's almost impossible for a national budget for most low-income or even middle-income countries. Even some of the wealthy markets that we are into will ration those and put a lot of restrictions on what patients can get those. That's kind of a more obvious one. I think some of the less obvious things are, especially with genetic diseases, that requires newborn screening, specialized genetic testing, enzyme assays, infusion centers, gene therapy hospital capacities, things that are often non-existent in a lot of these lower income markets, unfortunately. I think similarly, you'll get patients with rare diseases in these countries that die really young without ever getting diagnosed. So even finding those patients can be really difficult. So the market in that sense seems kind of invisible. Something else that we've seen even with COVID, right? cold chain logistics. So a lot of these biologics and gene therapies need ultra cold transport and storage, which is sometimes really hard to maintain in rural areas or tropical areas. Something that we've seen also with our CF medicines, I personally, I know that making one of our CF medicines, it seems really easy on the surface because it's a pill. But actually the post-treatment follow-up for our CFTR modulators in terms of following up for side effects, following up for liver monitoring, liver enzyme monitoring, that monitoring is actually very expensive and complicated. And you need that regulatory and safety and medical infrastructure in that country, which again, doesn't usually exist if they didn't have sort of screening for that particular rare disease to begin with. So I think rare diseases in particular can suffer from a lot of these other challenges. And then obviously the low patient numbers for some of these rare diseases, tends to sometimes be less advocacy or less specialized NGO involvement. We're really lucky with CF that there's a huge CF foundation that is very well funded and very, very active, but not all rare disease communities have that same sort of support globally as well. But I think, like I said, there are a few different models that do exist. There's obviously donation programs. Novartis and Merck have been, very good at this. Novartis provided free Gleevec, I think it was, for leukemia. And Merck gave ivermectin in really large quantities for river blindness in Africa. Gilead did a licensing agreement with the medicines patent pool to allow generics for its HIV and Hep C drugs for a lot of different developing countries. There's also tiered pricing models. Gilead did this for its Hep C drugs for lower income countries, so different pricing models for different countries based on their GDPs. So these models exist, but most of these examples I just named off are from the sort of mega pharmas, right? So I think for a company like Vertex, for example, we're only 6,000 employees, which is really small compared to some of those others. And the majority of our staff is still really on the drug development side of the business. So I think we need to really think of ways where we can partner with an entity that can maybe take on the manufacturing or supply chain elements and the local knowledge. So thinking more about a mix of partnerships between pharma, global health entities like the WHO or Doctors Without Borders that already have that sort of crisis infrastructure or low-income country development infrastructure. Again, it might seem straightforward because in our cases for CF, the medicine is a pill, but it's still that, life-threatening multi-organ condition that requires intensive monitoring and a lot of different types of specialists. So I think we need to really think beyond just it being a pharma solution. And there just really aren't that many examples yet. And especially for something like a gene therapy or a cell therapy, that infrastructure just isn't there. So I think my personal dream would be to see some sort of cost sharing, multiple manufacturer partnerships around sort of disease or platform clusters to build maybe, for example, for gene therapy, a gene therapy manufacturing hub, like one in Africa or one in India to support, the regions around those areas. And I think also there needs to be innovation in how payment models are done. I think we're already seeing this in the US. There's new payment models like outcomes-based agreements where a country can pay in installments only if the medicine works. We're seeing that a lot, especially in cell and gene therapies now. We're definitely going to have to do something like that for our type 1 diabetes cell therapy. So I guess to sum up, it's not just the price. I think it's really the entire ecosystem right now that is sort of missing, that I think really needs to come together for high cost rare disease innovations to come to low income countries.
Thank you,
Sorry,
Ms.
that was a
Bhaiwala.
very long answer.
Oh, thank you, Ms. Bhaiwala. Your comments around cystic fibrosis are particularly interesting. So our next question is for Dr. Ganem. So since the bird flu outbreak, what have we learned from other zoonotic diseases such as COVID, Ebola, among others, and how can the countries better prepare for an incident like COVID-19?
That's a good question. This is a good news and bad news situation. The good news is that we have learned quite a lot of academic things about pandemic infection. This book learning, however, has not always translated into public policy And I think the prospects for doing it are getting worse. So what have we learned? Well, we've learned a lot about, for example, airborne transmission during the COVID epidemic. When all of us were in medical school, we were taught some very simplistic ideas about how respiratory diseases are transmitted from person to person. We were taught that there are two modes, droplets spread, with which large droplets that are aerosolized from the respiratory tract fall within a few feet of the individual, and people only get infected when they're in contact with those large droplets. And then there was considered a whole separate mode of transmission that was called airborne transmission, in which much smaller droplets from deeper in the respiratory tree can hang around in the atmosphere for hours and expose people even after the donor of those droplets has left the room. We know, common colds are examples of the former, and things like measles and tuberculosis are examples of the latter. And it was considered this sort of very bifid kind of model. Well, of course, there was never any real logic to that. If you look at the particles that humans emit as they breathe, it's a blurrogram. It's a continuous, there's a continuous size distribution. The people who study this the most are engineers and physicists who've been studying aerosolized, the properties of aerosols and aerosol mechanics. And those people have very little contact with clinicians, and epidemiologists tend to look down on them. And in fact, they had a great deal to learn from one another. And now we have a much deeper understanding of this. And it turns out, and of course, it was always idiotic to think that these two modes of transmission are mutually exclusive. They can, of course, operate in tandem. But it's much harder to document the kind of long-distance aerosol transmission that comes from these very small so-called droplet nuclei. We've learned a lot about that, and we have a lot of knowledge about that now. And now we know that COVID certainly is transmitted that way. Influenza is surely transmitted that way. That's not to say that large droplets and close contact doesn't also spread infection. It certainly does. But we've been putting the emphasis on the wrong syllable. In things like COVID and influenza, airborne transmission is actually more important than immediate close contact, the kind of things that separating yourself by 3 to 6 feet from the next person in line at Safeway is designed to protect you from. So we've had a tremendous explosion of knowledge about that. And I think, but the question is, what use is that knowledge? And that the main thing that one derives from that knowledge is that for airborne transmissions of the type that we saw in COVID and to some extent in influenza, we need to be rethinking how we treat indoor atmospheres. We need to have, especially in places where children are in close quarters with one another, or where anybody is in close quarters with other human beings. We need to have proper ventilation installed, and that's expensive. And there's, the minute the COVID pandemic subsided or was declared officially over, all the impetus to do that expensive re-engineering of school ventilation, church ventilation, workplace ventilation, basically evaporated. And so I'm worried that all those things academically that we've learned about transmission, we haven't taken the next step to, effectuating that. So it's a good news, bad news situation there. We've learned an awful lot about infectivity. Infectivity, it turns out, is not a fixed thing. In COVID, and I think this is also true in tuberculosis and many other infections, infectivity is distributed in a sort of bell-shaped curve. But it turns out that in COVID, for example, 80% of the human infections are attributable to less than 20% of the people in the room. These are the so-called super spreaders. And we really need to understand that phenomenon better. Now epidemiologists are quite sure about that fact, but now we need to understand what makes a person a super spreader. There are some things we know. For example, vocalizing, especially singing, anything that causes your larynx to vibrate at very high frequency will generate large numbers of small particles that can stay airborne for a long period of time. This is why these church choirs are particularly dangerous, and they're singing in a small room. But there's also a lot of other stuff goes into this. Titers of virus in the upper and lower respiratory tract. We know that most viral stocks are made up predominantly of non-infectious particles that have small defects in the way they're assembled. They look normal in the electron microscope, but they're not infectious because of assembly defects. If you just count the number of genomes that somebody has in the upper respiratory tract, it turns out to be not a very strong predictor of who's going to transmit. We need to have more sophisticated ways for assessing infectivity. But these are all things that we've learned from studies of pandemic diseases in the past 10 years. There was certainly all of that was true in COVID, but the idea that super spreaders are disproportionately important in transmission was also true in HIV, a sexually transmitted disease. There are super spreaders of HIV. And we need to understand that phenomenon better. But I'm worried that all of that book learning that we have assimilated has not affected a lot of things in public policy. And much of what's going on in our contemporary society and in politics is operating to subvert everything we've learned and to prevent its translation in a really effective way so that we can have a more robust response to the next epidemic. Just look at the fact that now an anti-vax, one of the leaders of the American anti-vax movement is now the Secretary of Health and Human Services. And I just learned that our ability to make an RNA-based vaccine for bird flu. There's a large trial that Moderna was getting ready to conduct under federal auspices. Funding for that has just been suspended. It's being quote unquote reviewed. But I think in the climate that we're in right now, I'm very dubious about whether, I'm just wondering what the motivation for that suspension of funding was. And we've made it very difficult. For the other major thing that we've learned from airborne transmission is how effective masking can be, especially when it's universal, when masks are put on the infected people to prevent the spread of particles, as well as on the recipients to prevent them from inhaling them. Universal masking is very effective, but now it's become a political football, and there are schools. We have developed a culture war around the masking issue, and that's really deeply stupid and very very difficult. If you want to translate, hard-won, scientific lessons into public policy, to have that be happening in our politics and in our culture is depressing, to say the least. So it's a good news, bad news situation. We've learned a lot, but in terms of translating these into action, we've almost taken a step backward.
Thank you, Dr. Ganem. Our next question is for Dr. Kleyman. What part of the process to develop and test a new drug or therapy is the most challenging, and why? How much does funding affect experiments or research plans? Or said differently, does knowing that you have access to a certain amount of funding affect plans for a certain trial or experiment?
This is a good question, but a very complex one. It depends on what angle you're coming at drug development from. The whole process is exceptionally complicated, and my colleagues here have named a couple of issues. So you have the question of, how do you know your drug is working? What are you looking for? How are you testing it? This is where biomarkers come in, which is my particular specialty right now, and finding out, how does one biomarker, which is some kind of biological readout, be that a DNA signature, some kind of RNA signature, a protein, or a combination of them, how do they relate to a patient getting better or worse from your drug? And if you don't see a clinical effect of your drug, did it do anything? Did you hit your target? Did you hit your target correctly? Did you hit just your target, or did you hit lots and lots of different targets and you just don't know it? It's kind of like my boss always likes to give the example of one of those switches in your house that controls all the circuits. It says that the circuit is going to turn off the fridge. You go over there and you flip the fridge circuit. All of a sudden, the lights upstairs go off. Obviously, you have some kind of wiring issues, and you need to figure out where those wires actually go. So that's one thing, like, why is your drug working? The other thing that was hit on was how do you distribute your drug? What does your drug look like? What's the formulation? What does it need in order for it to get to your patients? Does it need refrigeration? Does it need some kind of injector unit? Do you have to train your patients to actually self-inject with some of the new biologic drugs that are coming out where you actually do self-injections? Or do they have to go to a clinic? All of these things play into how accessible is your drug? How many patients can you recruit? Are patients willing to go into your clinical trials to actually trial your drug if you tell them that they're going to have to take 40 pills in one day because you can't find a formulation that will give them only one pill or only one injection? So that's another complication. You also have the issues of what patients are you looking to recruit to your trials? Do you want to target your drug to only the very sick individuals, at which point any kind of response might be considered good, but if a person with low levels of disease comes in, they're going to have to suffer through a lot of adverse events or just dangerous side effects of their drug because you targeted it to a much sicker population that could tolerate such bad effects. So really the thing that I find the most complicated and the most fascinating is all of the departments within a big company that come together to make clinical trials work and to actually get a drug from the bench to the patients and onto the market. And we lose a lot of very interesting science along the way. We have hundreds, thousands of potential drugs going into our pipeline at the very beginning of clinical trials. And these are drugs that have been vetted. We have shown, say, in tissue culture that they actually hit their targets. We've shown in animal models that they can reverse disease or alleviate disease or whatever you're targeting. And then you start putting them into clinical trials and you don't really necessarily know what you're looking at all of a sudden because what you saw, the beautiful picture of your mice not having cancer anymore, all the tumors shrinking, you're not going to see that in your human population. You might see it in a subset of patients, but then the question is, was that your drug? Did they spontaneously go into some kind of remission? Did something else happen? And what are the side effects? And so seeing how all of that comes together, the actual operations of making this work is what I find the most critical, the hardest. And this comes into the funding question. This all costs a lot of money. You have to have a lot of professionals, high-end professionals working together and figuring out every little piece of this puzzle. We have, at AbbVie, we're huge. So we have a department dedicated to pretty much anything you can think of. We have a department that's dedicated to making syringes. And they trial various sizes of needle, various thicknesses. Viscosity of the actual drug matters for these people, as does how did you hold it? Do you have to keep it refrigerated? And if you pull it out of the refrigeration, is the substance going to crash out of solution? Do you have to shake it? Do you have to avoid bubbles? All of these fun questions that you never thought had to go into this, they do. And then we also have a full department that only handles shipment of samples and shipment of lab supplies from us or from our partners to all of the clinical sites. A clinical trial isn't just in one location. It's usually worldwide or at least across the United States. And so you have the logistics of actually getting your materials, your drug, as well as your test kits so you can get the results back. You have to ship it there, you have to ship it back, you also have to store it. And everything has to be well labeled because it's so easy to lose samples. So funding, yes, absolutely. We have to be very careful how much we spend. We set our budgets, we argue about them, we talk about them, we see who else has money that we can scrape up for various experiments. We align our goals across the company just so we can make sure that everybody's being able to use all of the resources at our disposal. And then we have to go and get all of this approved. So we have to be very careful what we choose. We have to make sure that what we're testing actually matters. And we have to prove it to multiple different experts with multiple different goals in mind. So I have to bring my plans for what I'm looking at biomarkers. I have to speak to the medical director. I have to speak to anybody who's working in, say, companion diagnostics, if they're working with us to make a diagnostic test. I need to talk to the financial department that we have it all. I need to talk to the legal department to make sure that I've gotten the right permissions for all of these samples and so on and so forth. So I hope that kind of answers the question as best as I could. There's a lot of moving pieces here.
Thank you, Dr. Kleyman. Our next question is for Dr.
Or
On.
can I inject just a thing here?
Oh, sure.
It's a very complicated subject, and I'd be interested to hear what Andy has to say too about this. But I just want to talk to the students who are, studying biology and chemistry for the first time and thinking about, what goes into drug development. And I just want to tell you how cool it is. That one of the coolest things about being involved in early drug development in the very beginning, setting up a screening 1 million, 2 million compounds, finding hits that appear to hit the target that you're looking at. And then comes a very complicated, interesting, really cool process that involves chemists, synthetic organic chemists, medicinal chemists who modify those molecules, retested by the enzymologists and biologists to see that they're getting more potent. Then, putting them into animals to see that they are active in animals and that they can be absorbed orally or have to be given intravenously. And then, the molecules undergo a tremendous amount of refinement during a multi-year process that involves crystallographers, computational biologists who are looking at the structure of this molecule bound to the enzyme and then suggesting things to the chemists about where additional interactions could be picked up. And you're a biologist watching this whole process. It's just so cool. It's very fascinating. And it requires that you be very scientifically literate, that you pay attention in chemistry and physics and make sure you have a good enough background in physical science as well as biology to be able to monitor all of that. And then comes, of course, all the integration to the clinical disease. What patients should this drug be designed for? What are their needs? How does all that square with all that molecular work that you're doing? It's a really fascinating process, but it is so cool and it's a lot of fun. And it is from a point of view of just pure scientific sophistication, just as sophisticated as what goes on at Harvard and Princeton, in labs that are considered more basic science, that are not trying to do this kind of applied work. I'd love to hear what Andy has to say because I'm sure he's still in the thick of it.
Yeah, thank you. Yeah, I think that really I agree 100%. Don, with what Don says is that it is a really cool activity to be a part of. And Marianna outlined how there are all these different groups of people, these teams who's developed a tremendous amount of expertise in doing what they do, and then being able to interact with them to collaborate to really form these overarching solutions to the whole drug discovery and delivery enterprise is an amazing organizational challenge, but it's also an incredibly wonderful thing to behold when it happens, and it really delivers for the patient. One of the things that I really preoccupy myself principally with is, as I mentioned, how do certain targets, how do they actually lead up or roll up to really being presented as a therapy, right? Could targeting this particular gene be a therapy for this disease? Well, often most diseases don't show up saying, target here. You have to actually really unwind the biology of it and say, well, blocking or promoting this kind of target is actually going to help a disease and actually in the end have a meaningful value for the patient. That it's a big part of actually being able to restoring function but also avoiding side effects as well. And so just navigating all those different components involves teams of people really working together. When it works beautifully, teams focused around the patients and what their needs are, what their interests are, and how they experience their disease. And being able to, I think, just like anything else that one would do in life, that meaning that comes from this enterprise really is about focused on the patient. And I think that one of the things that you can really do as a person who will, come from Andover and go on to really lead teams, smaller and larger, into this enterprise is really translating what the meaning of that value is to patients and being able to envision what that benefit is. This is the thing that actually gives everyone on the team meaning in their lives. It's why I get up in the morning before my alarm goes off every morning, is because I'm just like, okay, we've got these things to do today, and that kind of motivation when you have it, because I've met my patients and I know what their concerns are and I know what they do, but then if you can share it, and that sharing comes in the form of stories, that sharing comes in the form of the real meaning that such a therapy of being able to break through some of those limitations really brings to patients, is something that really ignites the motivations and leads teams, leads yourself, but also leads all the people around you who haven't met these patients necessarily. But you can still, again, being able to communicate that kind of importance really is the essence of why we do anything in life. That the importance of what you do has a really important driver of achieving with the accomplishment that you need to get hard things done, and then with the coordination and dedication it takes to go overcome the problems and finding solutions to really, really complex questions along the way.
Yeah, it's like a thing of beauty when you see these come together. When a project actually works, it's an amazing effort, and being able to be part of that story that you tell people of where you started and how you got to something actually successful. There's nothing else really in the world like it that I've seen.
Yeah, the culmination of every drug hunter's career is the approvals that come for the medicines that you've created or you've contributed to. Those are really the things that really define your life, and knowing something about the patients who you affected, whose lives you affected, are really just just creates an incredible sense of meaning to your life.
Thank you, Dr. Ganem, Dr. On, and Dr. Kleyman for that elaboration. It's certainly an incredible process and a good reminder that this is all patient-centered. So our next question actually goes back to Dr. On. So how have AI and machine learning algorithms impacted the drug discovery pipeline? And what are the consequences for discovery ownership? And are there ways in which you think AI has changed the pharmaceutical industry for the better?
Wow. Thank you for that very, very um, broad question. I think that I'm going to probably take the liberty of picking and choosing some of the components of that, and just someone panelists hold me to the fire if I've left out something really important. I'm really excited about the opportunities that AI have presented for us in industry, for some of the less obvious things, and still preserve and know that some of the most important, hard, difficult things still remain for us as human beings to really work on and do. I think that one of the just really straightforward things that probably come with your software, the software embedded in a new computer now that you might get, are the ways in which you can automate a lot of things. The way you can summarize this meeting into a set of bullet points, or the way you can collect data that's out there. And when we're talking about patient data, we're talking about very complex networks of data sets. They're hospital records, they are insurance claims, they are all these things that really are relevant to understanding how a patient experiences their health and their disease. And being able to then do any kind of hypothesis testing within that realm of understanding how a patient experiences their health and their disease is exquisitely tedious, difficult, and also involves a huge amount of data. So being able to scrub all that data in a much more efficient way, those are the kinds of things that AI is really, really well suited to. And I'm really excited to see us really being able to capture all that opportunity. Now, even better is that when you can then use it, collect all that data and prepare it and analyze it to some extent in which the usual ways of analyzing that data are possible. And then even in the project of just screening drugs, Don mentioned that you might have to screen millions and millions of compounds to actually find out what kind of compound might have the effect that you're looking for. Some of those might actually have to be from direct screening. You might actually have to apply an assay to it. But eventually, after doing several thousand of them, or maybe 100,000 of them, you might actually start to get to see the patterns and how certain structures and molecules may actually start to behave similarly to each other. And so you may not actually have to do all of those screens to actually help identify the major classes of kinds of drugs that you might want to focus in on further and later. And so then being able to then say predicting those drug properties, I look forward to seeing how we can use more advanced methods to be able to find those right ones, and then to optimize some of those medicines to be less toxic, or to optimize them to be better metabolized, or to be more efficient in binding their targets. There's one other part where there are a bunch of approved medicines, and being able to find applications for them really because of being able to notice patterns in which some medicines have been working in the population may also, again, be one of those ways of scrubbing the data, scrubbing the ways in which medicines are currently being used to be able to do that. Even more directly in my shop of designing then clinical trials to prove that this is a real thing. Some of the nuts and bolts of that are like the trial design, the protocols, the informed consent forms, all those things, just enormous amount of work that involves just elaborating some of those things. There's probably a certain amount of automation that's associated with that. The one thing that I think, and this is a controversial point, because in fact, some companies have made huge investments in this direction, which is to really try to figure out how individual targets correspond to their diseases. I think this is really the holy grail of drug discovery for me. Still the last bastion of where it takes really undefined complexity and understanding of targets and the disease processes, where being able to figure that out, you might be able to get some assistance in doing that, screening through targets and drugs that have, or targets that have certain characteristics that you're looking for. But in the end, there are a lot of these hypotheses where, again, that translation of a patient's life and disease into a pathway and into manifestations that you want to try to address with certain pathways, molecular pathways or cellular pathways. I don't know that there have been enough drugs out there and enough targets out there validated so that we have enough information for a machine to really figure out from those instances of discovered and approved and validated targets how to find the next validated target in the future. That, I think, is probably requires, we only scratched the surface right now in terms of the number of drugs and targets that are validated for certain indications in medicines. This is a very small number of drugs. And you need to feed AI many, many more instances of those kinds of applications to really get that kind of thing. Right now, it still depends on, all of you one day coming to understand these pathways, these molecules, these enzymes, and these gene targets, and then understanding the patients, understanding the diseases, being able to make those connections, being able to come up with more systematic ways to make those experiments to help clarify which of these targets and pathways might be the most productive. There's a lot of work for you to do, so get going. Have I left anything out major? Have I made any big gaps?
You did a good job. It's a difficult question.
Thanks.
I think I can add one smaller example. I think you touched on it a little bit in terms of like claims data. I think one area that we're starting to see more AI used kind of on the real world evidence health economic side is kind of when I was going back to one of my prior answers around sometimes payers, for example, don't know how to identify their own patients for a given disease in claims, especially for some of these rare diseases. We've seen in some of my prior work in consulting, we had clients that were working on drugs for diseases that didn't even have ICD codes yet. So physicians were kind of not sure, were miscoding them because there wasn't a code for that specific disease yet. So now that there are more data sets emerging that are sort of EHR, so like electronic medical record data linked with claims data, that EMR data typically was very unstructured, but something like AI can kind of comb through the physician notes better than we can, in terms of like SQL programming, and try to start, coming up with patient finding algorithms better than what we were doing manually as, 24-year-old consultants. So I think that's another area that we're seeing from our AI teams that they're working on for, again, very, very rare diseases where there just aren't examples that we can draw from, and we need to be able to find the patients to size the population and then start doing research on them.
Thank you, Dr. An, and thanks to Ms. Bhaiwala for additional details around the business elements of these AI components too. Our next question goes back to Ms. Bhaiwala. Although your role is focused in North America, you have spearheaded efforts in far-reaching areas of the globe and actively sit on boards that work with international missions. What's the common misconception about your role or the work of Big Pharma? What do you wish people better understood about its impact or challenges?
I would love to hear other panelists' answers to this question too, so I'll try to keep it short. I think I'm glad you asked this though, because I think it actually lends itself to more of a comment on career development that I can talk about later in one of your later questions. But I think there's a common misconception, at least that I had even in grad school, that going to biotech or pharma is selling out in a way. I think for myself, I certainly thought that for several years, and I came out of graduate school believing that that's what I was doing, that I was selling out, and that continued to be a motivator for me to continue a lot of that sort of service work and international work on the side. But again, now that I think I've been in it a few years and have gone through more of the processes, you really start to, and I think the panelists already touched this in some other answers, you really appreciate the vastness and the complexity that is drug discovery and access. And not even being a scientist myself, just being part of the organization, you see it. And it takes 10 to 15 years, $1 billion plus to develop just one drug. Like I think 90%, right, of clinical trials fail. Takes time, bureaucracy, money to do this type of research. Same with economic research, not even close to the same, but it takes time for these things. And I think more specifically on my role in health economics, there's a big misconception about the commercial side of biopharma, that it's purely focused on pricing and sales. I think there are certainly some actors in the industry that have made a name, in a more negative way. The opioid crisis is a great example of that. We obviously do have a pricing team, but they're one piece of a much, much larger puzzle. And I think one thing I didn't even expect until coming to the industry is that health economics in particular, and the commercial side of the business, it really actually involves a lot of us going back to the roots of public health and sort of being able to appreciate that in a different way that I wouldn't have before. So I think just giving a few examples, at least from Vertex, like in the last 2 years, we've come out with the first CRISPR gene therapy for sickle cell disease. Sickle cell disease, as many of you might know, extremely underserved patient population, disproportionately affects Black Americans. And so one of the projects, I didn't even work on this, but I got to hear about it, which was fascinating. But, we have a patient support team that works with a couple of different sickle cell foundations throughout the United States on different education campaigns. And one of the projects that they did was actually helping to educate the Black community about what it means to have sickle cell trait versus sickle cell disease, how to have those conversations with potential dates that you're going on, because for family planning and future planning like that. So they actually organized like a speed dating night for Black singles in Texas, which is one of the biggest hotbeds of sickle cell disease, to talk about sickle cell trait and do like a speed dating thing after that. You wouldn't necessarily think like that's part of, how to bring therapies to patients, but all of these different things have to happen or these medicines will never actually get to patients. Similarly, earlier this year, people might have seen in the news, we came out with the first non-opioid pain medicine in over 20 years. Don't need to tell you guys how the opioid crisis is a public health disaster. But at the end of the day, payers need to know, how are we going to get these patients into the healthcare system? How are they going to find these patients? How much are they costing the system now? And how is the medicine we're bringing as the manufacturer going to actually change anything, both from a clinical perspective, but also from a quality of life perspective and an economic perspective. And so I think that's a misconception that in our job, we actually really do. I think Dr. Ahn said that we need to understand every aspect of the patient journey. CF, cystic fibrosis, is a great example too, because I think for a long time, people and payers as well thought it was just a lung disease. It's actually a multi-organ disease that affects the lung, the pancreas, the GI tract, the liver. There can be a lot of different phenotypes of the disease as well, and it requires a lot of different coordination with different physicians, and a lot of different complications that ultimately result in hospital bills. But it's not just the cost to the patient, it's the cost to the caregiver. So the parent of this little child that's having an exacerbation and needs to be rushed to the hospital, that adult parent is missing hours from their job, which has a downstream effect on the economy. And so we have data that we do research on to show how caregivers of patients with CF, how many of them have to switch from full-time to part-time or part-time to no job to take care of their child. And again, that's ultimately resulting in a loss of a productive contributor to the economy. Sounds impersonal, but at a patient level, you know, if I'm a parent and I have to rush to the hospital, there's a lot of different things going on. And I think also important is kids with CF before modulator therapies, they weren't able to plan for the future since the average age of death was well below 30. Now they can go to college, manage their disease on their own. They can get married, they can have children, continue to, you know, reproduce the human race. So this is all data that we can slowly see over time and build into what we call societal models to show governments, like how on a societal level are these medicines changing the dynamics of a population. So all of these different aspects of, I think, what we would traditionally think of as more public healthy also play into our jobs on the commercial side of the business. So again, really all going back to what we were saying about deeply understanding the patient is essential for us to do our job well and ultimately get the people who pay for these medicines on board, because you can have an incredible medicine, an incredible feat of science, but if the payer is not on board, it will never get to patients. And that's just in the US. And that's where the buck really can't stop. And I think there have to be people within each company saying, okay, great, it's in the US now, it's in Europe now, how do we get it to everyone else?
Thank you, Ms. Bhaiwala. Our next question is for Dr. Ganem. As a medical doctor who has worked in the academic world for 2.5 decades and then decided to shift to the private sector in pharma, how did you leverage your background to make such a significant career shift?
Well, I'm sure Andy has a similar story to tell, and you should ask him this question. For me, there was a big progression over the course of those 25 years. When I started out, you know, I had a BIFID career. I had a lab of anywhere from 12 to 15 people. And I also served on the clinical service, seeing patients with infections in the hospital 2 to 3 months a year. Those two worlds are very different worlds. Even though they seem, you know, lay people think that medicine is very scientific and that biology has a great deal to do with medicine. In fact, these are very different worlds. They require different strengths and they have different predicates, which is why very few people continue to do both of those things together for more than a decade or so after they graduate from medical school. By the time most physician scientists are in their 40s, they've usually stopped seeing patients and tried to just go back and retreat to the comfort of the laboratory. I had a kind of funny evolution that went in an opposite direction. I got more and more interested in medicine as I got older, and I wanted more and more for the science that I did not to just be about a paper in Nature or Science, but for it to be about something having to do with human disease, so I never stopped attending in the hospital. I never stopped working in the hospital. My last day in the hospital was the week before I started Novartis. You know, I was still doing 2 to 3 months of clinical service in the hospital. right up until I was 60 and left for Novartis. So it was an unusual trend for me, but you know, that's the way it played out for me. I loved medicine and I loved working in science, but increasingly I found just writing papers, you know, just publishing things that other scientists found interesting was just not enough for me. I really wanted to see it evolve in a direction where my work could have an impact on the practice of medicine and not just, you know, be illustrative of things that somebody else would ultimately apply to medicine. So, you know, and this came to a head during the AIDS epidemic in San Francisco in the 80s and 90s. You know, I'd been working on hepatitis B, and my work there was so abstruse. It was just pure biochemistry. Um, and you know, I was asked to give medical grand rounds on hepatitis B to my fellow clinicians, and I realized that nothing I was working on in the lab would be accessible to them and would be interesting to them. And I decided that it was time to change fields and do something that would be more related to human disease, and that the work I did in my own lab could be explained to a practicing physician in terms that he would understand, or she would understand. So I started working on looking for the virus that causes Kaposi sarcoma, which at that time was the #1 tumor of AIDS patients. So I was moving in the direction of wanting to do more work that could be directly impacting patients. Around the same time, I was being asked to serve as a consultant for several biotech firms. Chiron, it was a company called Tularic that ultimately got bought by Amgen. I was on their scientific advisory board, so I started spending more time with industrial scientists. And I realized, you know, what an extraordinarily high level of scientific work was going on in those companies, and how sophisticated it was. But this was work that wasn't about discovering something brand new that could be published as a manuscript, but that took what we had just discovered and turned it into a therapy. And I really became very enamored of that. And as I ultimately served on the scientific advisory board at Novartis for 7 or 8 years. I got to know the staff there very well. And I realized that this would be the next step for me. And there's one other thing I want to say here about, you know, the academic world is a very different world from the world of industry. It rewards different behaviors. It has different predicates. It serves different purposes. You know, the purpose of an academic scientist is to discover new things, whether they're of practical value or not. The purpose of an industrial scientist is to make a product, okay, is to make a drug, a medicine, a vaccine. And those are very different worlds. The enterprise is organized in very different ways. Science in a university is a cottage industry, and your lab is the cottage, okay? Whereas in industry, it's a team enterprise. So academic work tends to prioritize individual achievement, and the rewards are handed out to those who are perceived as leading or you know, being primarily responsible as an individual for an achievement. Whereas in a company, the goal is to make a product, and since no one person has all the knowledge that it takes to do that, it's about harnessing teams, each one of which has a lot of expertise: crystallographers, enzymologists, virologists, epidemiologists, clinical investigators, pharmacologists, toxicologists, and the whole thing. No one person could know all of those things. But when I got to Novartis, I realized it was my privilege to lead those enterprises and to learn all that stuff. And it was just tremendously cool and a lot of fun. But there's one last thing I want to say, which is that the study of medicine really informs a career in industry in the following way. You know, when you're a scientist, you can spend 8 months, a year, 2 years working on a problem, you know, trying to nail down the splice junction of a minor alternatively spliced RNA, and you won't say a word to your colleagues until you're fully sure what the answer is there. And that could take a year or 2 years to just define that small problem. Then you go up to the wards and you see patients who have septic, you know, extremities and bloodborne bacteremia. and you look at the affected extremity, you measure a few simple lab tests, you walk out of the patient's room, and the surgeon is standing there, and his question to you is, should I amputate? So that's the real difference between a career in science and a career in medicine. In science, you don't say a word until you have nailed down all of the variables around a very small question. In medicine, you're asked to make gigantic decisions of great human importance based on extremely fragmentary information. And the skill sets required to do each of those things are different, and they often don't translate well one to the other, which is why there's so much tension between being a laboratory scientist and being a practicing doctor. But if you can understand those differences and master them, it puts you in a very good position when you come to industry, because when you get into industry, you're also going to find you're going to have to help those crystallographers and chemists who want to work with that level of precision. But you also have to deal with all the other uncertainties that physicians have to deal with. You're going to have to make a lot of decisions of great magnitude based on fragmentary information. And there's no better preparation for that than bedside medicine. Okay. I remember my first week at Novartis. I had been on the job for less than 2 weeks or so. And I went to a big meeting with the corporate leadership. And at that time, there was a lot of interest in producing new medicines for hepatitis C. And Novartis was interested in the possibility of acquiring potential compounds from small biotech firms. And I walked into a meeting, and after a certain amount of discussion, everybody turned to me and said, well, okay, Ganem, what do you think? Should we spend $30 million to bring this molecule in? And, I'd never spent more than $1 million on anything in my entire life. I mean, in academic science, if you can raise $1 million to run a lab, you're doing very, very, very well. And somebody comes in into my second week on the job, says, should we spend $30 million to bring this molecule in? When I wasn't even sure I understood all the parameters around that decision. But there's no better preparation for that, for making big decisions on the basis of fragmentary knowledge than if you've practiced medicine for 10 years or more. So it is possible to do both of these things. And once you're in that position, you realize that it's incredibly gratifying to be able to do that, to be in the service of where science is not in the service of just a cool paper in Nature, but the development of a product that's ultimately going to make, and change people's lives. And so I don't know, that's my take on it. I'm sure Andy and Marianna have had their own journey in this direction.
Thank you, Dr. Ganem. Unfortunately, we only have a few more minutes together, but our next question is for Dr. Kleyman. When you were a student, you worked closely with Dr. Hagler on a molecular biology research project at Andover. In fact, I am currently taking Bio 600, allowing me to explore my own research into the combined effects of Ozempic and BPA. Can you briefly talk about your project with Dr. Hagler, what it taught you, and how that fueled your interest in research?
I would love to. My experience with Dr. Hagler, it was the very first lab I'd ever worked in. And when I started, I didn't want to ever leave the lab once I got in there. We started very basically. My project was to purify a protein from vaccinia virus, which is similar to chickenpox, a little bit safer. And that's all I had to do. I had to take that gene, clone it, purify it, make it protein, and purify the protein and get enough protein to show that I have protein. At the time, we didn't really have all that many materials, so we had to find the sequence. We ran a giant silver sequencing gel, which I don't think anybody ever does, but they're like, I don't know, they're about like 2 feet long, and you attach like alligator clamps to it and push a bunch of electricity to it. Dr. Hagler was the one who had to do it because we were legally not allowed to come near the closet with that electrical device for fear of electrocution. We stained it with the silver stain, which splashes, and everywhere that it splashes, it leaves a little black dot. So the day after that in the lab, even though, you're wearing gloves, you're wearing a lab coat, somehow that stuff gets everywhere, and you come out and you're like, I didn't remember having this many freckles on my arms before. They're not freckles, they're just silver stain. And then hours of sitting there, waiting for columns to drip through, waiting for things to happen, finding out that you forgot to put an enzyme into your reaction, and so you got absolutely nothing and you have to do it again. And all of this sounds exceptionally tedious, but it was so much fun. It was so much fun to just work through a protocol, figure out what this is, put together the little pieces, and actually come out with something real. I did end up purifying a protein. I could show it in a tube. I could look at it on a gel. I could also play with the newly coming out 3D modeling programs that allowed me to look at that protein, see where it might be binding to things, look at the little deep pockets, see how the amino acids played with each other to form a shape. And just being able to do that, being able to tinker and being able to guide my own way through questions that came up as you do the work, that's really what fueled me in the lab. What was difficult was choosing an area to be interested in, because I wanted to know everything. I wanted to play with everything. I wanted to touch all of the proteins. I wanted to play with all the models. I wanted to do all the projects. And that's really not how you choose a graduate program for yourself. So having to actually learn to temper myself, to focus, to decide what is actually important, what is my question, and where do I want to end up, that was the hard part. But the love for it, the enthusiasm, just the wide open field of exploration and finding things that nobody else has ever seen, and you're the first one who could potentially see it, that was the thing that drove me. Everything else that happened was just life, and where it takes you is really, it's what you need in life, what you want from life. But the scientific exploration has never left, and it's just grown into something that I can apply to everyday life. I do not want to ever lose this sense of awe, the sense of how tied together the world is, and the desire to find out more about it, because there's always more to learn.
Thank you, Dr. Kleyman. So we just heard from Dr. Kleyman on her work with Dr. Hagler as a student, but wondering if there is a specific faculty member or class that you look back on Andover that helped propel you into scientific research or ignite your interest in the pharmaceutical industry. We can do this as a fire round to keep it brief, as I know we're running a bit over. So maybe we could start with you, Dr. An.
Wow. Okay. Just off the top of my head, English 10. It was called English 10 back then. Homer's Odyssey. Telemachus rises to meet the historic needs of what he has to do. He has to go and find Odysseus, and that process is one in which you require a mentor, and it's a long journey. The others is History 35. I think that people in history arise to meet historic events in great ways and small ways. And for me, then, my transition from academia to industry came from the discovery of a historically significant target and the validation of it for being able to work in an unexpected way. And for me to then take that and carry that forward into insights into how it could work in patients was an important one that I felt the need to do that for. I felt that it's hard to do. And I think you need the right person in the right room to represent the patient's voice in the end, to make those new indications possible. It did lead for me to be able to lead a team to a new indication for a medicine that had a disease that had never had a medicine for it before. So that first indication opportunity was something that was very important to me.
Ms. Bhaiwala?
Sure. I mean, it's embarrassing. I forget how the classes were numbered, and I'm the youngest one on the panel. But it was, I think, Religion 100 or something. Mr. Andy Huzio, Dr. Huzio. He had us read Viktor Frankl's Man's Search for Meaning, which I've since read like 5 more times. And I think it's that famous quote that he attributes to Nietzsche, that's he who has a why to live can bear almost anyhow. And I think that was really the first time I started thinking very intensely about how I can find a reason or purpose for myself. And I think at the time I was really passionate about a lot of different social justice issues, global issues, what have you. And it took me a while. I think I'm still figuring out, but I think I'm finally slowly arriving at a place where I think I can merge sort of some of the burgeoning expertise, I'm still earlier on in my career, but expertise in biopharma and my place in biopharma, merge that with the passion I have for global health and social justice. And maybe that's my purpose. And so that's kind of how I'm thinking about my career going forward, is how can I maintain that passion in this corporation, as you call it. But I think as we've discussed over this evening, it's definitely more complicated than that. So I think just being an example of someone who's continuously raising their hand and saying, hey, this is what I want to work on. This is what I care about. How can we make this happen within this framework?
Dr. Ganem?
Oh yeah, Andover was a very different place in the 60s than it is now. When I look at the curriculum that exists in that place in Andover now and see how many opportunities, scientific opportunities people have. I mean, this Bio 600, that didn't exist back then. There was one biology course, one chemistry course, and one physics course. And there was, and the student body had rather little interest in science. It was some people, there were requirements, but it wasn't like it is today, where you can study synthetic organic chemistry at Andover, something you couldn't do until you were a sophomore in college in most places back in the 60s. That there's, astronomy, there's plant biology, there's all kinds of highly specialized electives in engineering, sensibilities and computation. None of that existed back then. But I was very strongly influenced by the biology course that I took my sophomore years, the Introduction to Biology, because the book that it was based on had been written by an Andover faculty member named John Kimball. John Kimball had taken a year off to write a book, and that book was one of the first textbooks in the 60s that discussed biology the way we think about it today. It started from the structure of DNA and the nature of the hereditary material and the nature of proteins and enzymes. Before then, biology was just a rote exercise in taxonomy, and it was divided into zoology and botany. And that when I got enrolled in the course, I was expecting that was what it was going to be. And I was amazed to discover it was really a course about the chemistry of living things. And I was so turned on by that I knew right then and there that I was going to go and major in biochemistry. And I hadn't thought about being a doctor, but that I was going to somehow be committed to life sciences. So it all began with that book, which was written by this very extraordinary man, John Kimball, who was not my teacher in the course, but we used his book. He went on to leave Andover, get a PhD in immunology, and wound up becoming a faculty member at Tufts, teaching immunology at the college level for the rest of his career. A very interesting man. But he certainly changed my life. That was how I got into this line of work.
Thank you all for such thoughtful responses. On behalf of the entire Andover community, we are beyond grateful to all our alumni panelists for their time tonight. Marie Curie, the first woman to be awarded the Nobel Prize in 1903, for her contributions to physics and chemistry, famously said, "Nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less." This evening inspired so many of us as current Andover students, the alumni community, and beyond, to embrace the desire to understand the unknown and leverage science to improve the world around us. We are so grateful to hear about the pharmaceutical research and impact that you all are leading since leaving our shared home in Andover. I will now turn it to Mrs. Savino to close today's program.
Thank you. Sorry, my power of unmuting was taken away from me, but given back so I can close out the evening. Thank you. So first, thank you to our panelists. You have made time in what is clearly a very busy life, professional life, to share your journeys and your perspectives. And I think it's clear by how many folks are here tonight and folks hanging in that I think this is a conversation we would love to continue another night. So may I in real time extend an invitation to continue this conversation, and actually a warm invitation to come and do it in the Gelb Science Building at your convenience. listening to the paths that you all take. I appreciate the bumps and the curves and the differences. It's a great message to our students, particularly those who have shown up tonight with numbers 25 after their names. They are in the last few weeks, they are in their senior spring, and they are leaning in here this evening, making time for it. And one of the things that is always an objective of the alumni office is to show that the path is quite curvy and quite bumpy, and that hopefully Andover has instilled that resilience to travel all of it, and to be open for it. And the 4 panelists this evening certainly demonstrated that. So to everyone who has stayed on this evening, thank you so much for being in community. And thank you to our stars and to our panelists. We will certainly see you soon. Take care, everyone.
Thanks.
Thank
It was a
you.
pleasure.
Thank you.
Bye, everyone.